The breathtaking Denisova Cave is nestled near the Altai Mountains in southern Siberia, Russia. Once a refuge for an 18th century hermit, this mystical cavern has revealed secrets that challenge our understanding of human ancestry. In 2008, a group of awe-inspiring archaeologists from the Russian Academy of Sciences unearthed bone fragments of an ancient being within its depths. However, it wasn't just any ordinary discovery. These bones didn't belong to Neanderthals or Homo sapiens, but rather to an entirely new species of human, the Denisovans. Delving into the enigma of the Denisovans, we quickly realize that we know surprisingly little about these elusive beings beyond their DNA sequences. While they are believed to have roamed the Earth as far back as 217,000 years ago, only a handful of viable Denisovan bone specimens have been discovered thus far. Merely 5 of these precious fragments have undergone analysis, yet the scientific community was in for an astonishing revelation just a few years later. Little did we anticipate that a groundbreaking discovery awaited us. Not only were these ancient humans Denisovan, but one particular find unveiled an extraordinary intermingling between 2 distinct human species, shattering preconceived notions of human evolution. We ponder the mysteries of why Neanderthals and Homo sapiens managed to successfully interbreed, while mules, those fascinating equine hybrids, find themselves rendered infertile. To unravel this riddle, we must delve into the realm of DNA. You see, a horse boasts 64 chromosomes, while a donkey carries 62. When a mule is born, it inherits an odd number, 63 chromosomes, a genetic code that's considered defective. DNA craves balance, desiring an even number of chromosomes derived from both parents, precisely 50% each. Fertility emerges as the key to successful interbreeding, granting us a glimpse into the wonders of nature. While mules may be barren, numerous other species can produce fertile offspring. Take, for example, the majestic liger, a magnificent blend of a tiger and a lion. These creatures share a genetic compatibility much like many primates do. It stands to reason, then, that Neanderthals and Homo sapiens were capable of fruitful unions. Scientists hypothesized that Denisovans also engaged in interbreeding with other human species, regardless of whether their offspring thrived or remained infertile. However, concrete evidence to support this hypothesis remained elusive until 2018. Allow us to transport you back to the year 2012, where Russian archaeologists embarked on another expedition within the hollowed Denisova Cave. Excitement filled the air as multiple bone fragments emerged from the earth, eagerly awaiting identification. A staggering collection of 2,000 bone fragments was assembled and sent to a lab, where they lay untouched and unexplored for several years. But fate had a remarkable twist in store for us. Fast forward to 2016, and enter Samantha Brown, a brilliant MSc student at the University of Oxford, entrusted with the task of unraveling the mysteries concealed within these fragments. Engaged in the meticulous process of DNA testing to determine the origins of each bone, Brown stumbled upon a revelation that left her breathless. Amidst this vast collection, measuring a mere 2 centimeters, lay a bone like no other, a human bone. Aware of the cave's historical significance in harboring human remains, Brown's curiosity was piqued. She delved deeper, and what she uncovered was nothing short of astonishing, causing her initial disbelief. This minuscule fragment, it turned out, bore testament to an extraordinary outcome of interbreeding, an individual who embodied both Neanderthal and Denisovan ancestry. If Brown's findings were accurate, this would mark the first concrete evidence of first-generation breeding between 2 distinct human species. Eager to validate her groundbreaking discovery, Brown promptly contacted the head of her department. The bone fragments embarked on a journey to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. There, armed with cutting-edge technology, experts embarked on a meticulous examination of the bone fragment, ultimately confirming the veracity of Brown's astonishing findings. The surprise wasn't limited to just Brown. Picture this, a team of brilliant paleontologists, Viviane Sloan and Svante Pabo, delving into the world of ancient genomes. Their mission? To analyze the DNA sequences extracted from those ancient bones, meticulously tease out the connections between each species. To their utter astonishment, both researchers verified Brown's claim. Yes, that bone was indeed the extraordinary find they had been hoping for, a Denisovan Neanderthal hybrid, a true marvel of nature. No one in the scientific community had anticipated such an incredibly rare discovery. While it was hypothesized that Denisovans and other ancient humans interbred, finding concrete evidence so swiftly was beyond anyone's wildest dreams. However, the DNA analysis merely scratched the surface of this captivating tale. Who was this ancient human, and how did these two distinct species end up coming together in the first place? Delving deeper into the bone fragment, Sloan and Pabo unraveled more intriguing details about this ancient individual. Their investigation revealed that the bone belonged to a young girl, approximately 13 years old, as estimated from the width of her bones. Radiocarbon dating suggested that she lived some 90,000 years ago. The initial bone fragment, examined by Brown, turned out to be a tiny piece of her finger. In honor of the cave where she was discovered, the researchers named her Denny. The researchers embarked on a quest to uncover Denny's lineage by meticulously analyzing the DNA sequences. Their efforts paid off, revealing that her mitochondrial DNA hailed from a Neanderthal. Since mitochondrial DNA is inherited from the mother, it meant that Denny's mother belonged to the Neanderthal lineage while her father was a Denisovan. Nonetheless, the scientists involved couldn't fully wrap their minds around the significance of their findings. Doubts still persisted, urging them to seek further confirmation. To ensure the utmost accuracy, the bone fragments were sent to the esteemed Francis Crick Institute in London for a third analysis. This ancient human represented an incredibly rare gem, an individual born of the first-generation interbreeding, with both parents being fully Neanderthal and fully Denisovan. Such a discovery had eluded researchers until now, even in the case of first-generation Neanderthal Homo sapiens interbreeding. Denny's lineage may be well established, but when it comes to the details of her life, it's an enigma. Adding to the intrigue, little is known about Denny's paternal family history. The discovery of Denisovans is still relatively recent, leaving scientists with much to unravel regarding their historical significance and their connection to modern humans. To shed light on how Denny's existence came to be, we must explore the possibilities. Interbreeding between species is only possible when they inhabit the same geographical area. For instance, ligers, the offspring of lions and tigers, do not occur naturally in the wild because these animals live far apart. Denny's situation is similar, as different groups had to traverse long distances before mating could occur. Another intriguing possibility is that Denny was part of a Neanderthal Denisovan colony. This theory could explain the scarcity of Denisovan bones discovered by archaeologists. Harris remarked that he could count the number of pure Denisovan bones that had been discovered on one hand. If this is indeed the case, it suggests that there were few pure-blood Denisovans remaining. Their genes might have gradually mixed with the Neanderthal lineage and eventually faded away. However, it's important to remember that this is merely a theory. Denny is not a pure hybrid. Her DNA reveals a mixture of Denisovan, Neanderthal, and other hominin genes. The tests indicate high heterozygosity in Denny's genetic makeup, which refers to the amount of genetic variation present in an individual. For example, if two cousins have a child, the child would have low heterozygosity. On the other hand, if two individuals from different countries and ethnicities have a child, the child would have high heterozygosity. So why does Denny exhibit the later? Richard E. Green, a computational biologist, sheds some light on this mystery. During an interview with National Geographic, he describes Denny as being heterozygous out the wazoo. In other words, Denny's parents hailed from geographically distant areas and rarely engaged in interbreeding. Denny's DNA sequence reveals a closer relation to a Neanderthal from Croatia dating back 55,000 years, rather than to Neanderthals near the Denisova Cave who lived 33,000 years ago. This raises the question of how her parents could have possibly met. One possibility is that Denny's parents crossed paths in the cave itself. Most Neanderthals dwelled in Eurasia, while Denisovans roamed Siberia. The Altai Mountains, where cave discoveries were made, lie between these two regions. Svante Pabo theorized that any Neanderthal residing west of the Urals would never have the opportunity to encounter a Denisovan in their lifetime, due to their close proximity. If this hypothesis holds true, it raises the intriguing prospect of modern humans possessing Denisovan ancestry. Denny's existence challenges our understanding of human evolution. The discovery of her remains suggests that interbreeding between species might have been more common than previously believed. Prior to 2018, scientists had only studied 23 ancient human specimens, with only 2 indicating signs of interbreeding. This scarcity led experts to consider it a rare occurrence. However, Pavo stated that Neanderthals and Denisovans might not have had many chances to meet each other. He further suggested that when they did come into contact, their frequency of mating must have been higher than previously assumed. Denny's story inspired researchers to re-examine other ancient human specimens, such as OACE-1, a 42,000-year-old lower jaw, which upon closer analysis revealed a small amount of Neanderthal DNA. These findings challenge the notion that interbreeding was infrequent and suggest a more complex web of genetic exchange among early human populations. Paleoanthropologist Chris Stringer proposes that Neanderthal-Denisovan pairings offered evolutionary advantages. Genetic diversity allowed humans to adapt and survive more effectively. Additionally, both humans and animals thrive in larger populations. By forming stable pairs, mates could increase their offspring's chances of survival. Due to frequent migrations of ancient human groups, encounters with other species were inevitable. It is within this context that Denny's existence becomes plausible. Interbreeding also provides a fascinating theory for the extinction of the Denisovans. Pabo suggests that rather than violent extinction, the Denisovans might have become absorbed into modern human populations. With each interbreeding event, the genetic contribution of the Denisovans would have diminished over time. Consequently, their species may have dwindled due to intermixing. If this hypothesis proves to be true, then the Denisovans played a crucial role in the development of modern humans. However, like many other ideas, this theory remains unproven. Denny's story, along with other remarkable discoveries like OACE-1, unveils a complex tapestry of human evolution. It challenges our preconceived notions, pushing us to reconsider the frequency and significance of interbreeding between early human species. As researchers continue to uncover new pieces of the puzzle, our understanding of our own evolutionary history continues to evolve. And that's been it. If you enjoyed our efforts, then make sure to subscribe to the channel and press the bell icon for more content like this. We'll see you in the next one. Until next time, ciao!